Thanks for that very nice introduction, and thanks to everyone for coming here. It's great to be here at Nimbus. Today I'm going to talk about producer-grazer models that have been developed under this theory of ecological stoichiometry, and that's a way to include food quality into models. So to start off, I'm going to start with a motivating example to kind of introduce why quality is important and some of the consequences you might have if you neglect to include them in some ecological models. I'll then introduce this theory of ecological stoichiometry, which provides a framework for including food quality into the models. I'll then go through the formation of a producer-grazer model that includes food quality. And that's done in 2 steps. The first step is this LKE model. This was actually done before my time, where they include nutrient deficiencies into a producer-grazer model. So the effects of having low nutrient levels. And the work that I have done a lot of my work in, the second part of that is looking at food quality in the sense that when you have excess nutrients, what that does to food quality and how that affects grazer dynamics. Then I'll end with further applications of this framework. A lot of the work I'll be working on here as a postdoc. So here's Lodka-Volterra's producer-grazer model with logistic growth, the Rosenberg-MacArthur variation. I'm not going to spend a lot of time here because I'm sure we've all seen this many times before. I'm just going to recall that this X is the producer population, Y is the grazer population. And the only thing I want to mention here is a question about units. What are the typical units of X and Y? And that can depend on what model you're looking at. X is the biomass of the producer. Sometimes they look at it in terms of number of individuals or a density. So for this talk today, I'm going to be looking at this in terms of a carbon density. So the density of carbon in the producer population and the density of carbon in the grazing population. Here, these models are a producer-grazer system. They've been parameterized for a system of algae and Daphnia. So X is our producer. Here's a Cenodesmus, a green algae. Again, X is, the algae population density in terms of carbons per liter. So we're looking at units of carbon here. Y, our grazing population, that's going to be a Daphnia. Here's a Daphnia magna. You can see, or you can kind of see on the slide, the green gut here is filled with algae, so it is grazing on that green algae. Again, Y is going to be our Daphnia population, our grazer, and it's in units of carbon per liter also. So going back to the model, looking at this in terms of these new units, here we're looking at the amount of carbon in this trophic level, the algae population, and the amount of carbon in the second trophic level, the Daphnia population. And this model is essentially just modeling the flow of carbon as it goes between these 2 levels. I also want to note this carrying capacity here, K. That's the producer carrying capacity. That's going to have the same units as X. So this is a carrying capacity in terms of carbon as well. When we're talking about algae, they do photosynthesis. This is going to be related to the light intensity of the system. So here's some data collected by Urabe and others in 2002 of the same system. So here they have green algae and Daphnia. And they gave different light treatments for these. So under low light, you have this first panel right here. This dotted line is the algae population, and it's measured in terms of carbons per liter. So again, the same measurements that we have for our model right now. This solid line here is the Daphnia population, again in carbons per liter. And under low light, you see they're both kind of existing. It looks almost like a steady state. And that model I showed you can capture these kinds of dynamics. Turn light higher. You see what happens over here. You have the algae population increasing and then decreasing. The Daphnia population here during this similar. It looks like those classic predator-prey cycles we've seen. That's also something that can be predicted, and those dynamics can be captured by that model I showed you. Things get interesting when they turn up light very high. You see in this third panel of data here, the algae, again, it does photosynthesis, so it's soaking up the sun. It's doing very well. You see this high algae population density here. But despite the high food abundance, the Daphnia population remains low, and it's unable to thrive. So this is an example of when that Lodka-Volterra model is going to break down and fail to make correct predictions. And it's an issue of food quality. So that third set of data you see in all these slides, the open circles connected, that's the algae phosphorus to carbon ratio. So the amount of phosphorus in the algae. This can be thought of as food quality. So under low light, notice the algae population has kind of a low density here. But if you look at the phosphorus to carbon content of the algae, it's up high. It's high up here. So it's kind of good quality food, and the Daphnia population does well on it. Look at the highlight example. Again, you have this high abundance of algae, but look where the phosphorus to carbon content is. It's down here. So it has a low phosphorus content. of food, which is bad quality food for the Daphnia, and they're unable to thrive. So they have a lot of food. It's high quantity, it's just low quality. They can be eating a lot, but they're essentially just eating a whole lot of junk food, and they're unable to do very well as a population. So this brings to point the kind of obvious fact that things are composed of more than just carbon. We're composed of several chemical elements. So the approach of just thinking about carbon as a single currency model, like the log of Volterra model, might lead to some pretty serious consequences. And maybe a multiple currency approach would be better. Okay. So this theory of ecological stoichiometry is kind of based on that idea. Here's a book on it written by Bob Cerner and James Elser in 2002. This is a study of the balance of energy and elements and how they interact in ecological interactions. Okay. It provides constraints that we can put right into our mathematical models. For example, that producer-grazer system, the algae and Daphnia we were just talking about, instead of just thinking about things in terms of carbon, where all the organisms are made of carbon, think of them as made as two essential elements, carbon and phosphorus. Then you can look at the phosphorus to carbon ratio of each of those organisms and bring food quality into the model. And that's how they did it here in this LKE model. So the LKE model, written by Irakli Lolads, Yang Huang, and James Elser in 2002, they set out to introduce food quality into the log of Volterra model. This is the same log of Volterra model. I'm going to show you how they formulated it. Okay? So they set out to introduce nutrient deficiencies and how that would affect these organisms and how they grow. So the two terms they changed are the two terms that are highlighted here. The first one is this carrying capacity for the algae or producer population. If you have a population of algae, its carrying capacity here is defined just in terms of carbon. But if you have limited amount of phosphorus in the system, the algae might be limited by the phosphorus. So they're going to change this to incorporate phosphorus limitation on algae carrying capacity. Same line of reasoning, they have to change the the grazer's growth rate. The grazer's growth might be limited by carbon, as it is in this model now, but it might be limited by phosphorus. So they switched those two terms into what you see here. So they replaced them with minimum functions. So looking at this new carrying capacity is the minimum function of two inputs. The first one is K. That's the carrying capacity from before. It's a carrying capacity defined in terms of carbon. And it's going to be related to the light intensity for this system. That second input in that minimum is going to be the carrying capacity defined in terms of phosphorus. In order to do this, they made a few assumptions. They assume that the total amount of phosphorus in the system P, right here, is constant. They also assume that all the phosphorus in the system is divided into two pools. It's in the algae or it's in the Daphnia. So they don't allow phosphorus to be out in the environment. So the total amount of phosphorus in the algae is going to be given with this expression right here in parentheses. So that's the total amount of phosphorus in the system minus the phosphorus that's in the Daphnia. So they assume theta is the grazer's or the Daphnia's constant phosphorus to carbon ratio. Dividing that by a minimum phosphorus to carbon ratio gives you the carrying capacity now defined in terms of carbon. Okay. They changed the grazer's growth rate here, the term highlighted in green. You now see another minimum function here. If this minimum is 1, that's going to be grazer's growth limited by carbon. It's going to be the same equation as before and follow the log of Volterra expressions. However, if it's going to be limited to carbon, the minimum is going to take this value right here. So what this value is, it's comparing the P to C ratio of the algae with that of the Daphnia. So the Daphnia has a constant P to C ratio of theta. The algae has a varying P to C ratio here expressed as Q, sometimes called the quota. Okay? So here, that's where the food quality comes into play. The algae comes of varying quality, as it has a different phosphorus to carbon content. So if you're a Daphnia, you have a constant phosphorus to carbon ratio of theta, and you go and you eat some algae. You look at the phosphorus to carbon content of the algae that you're eating and compare it to your own. If it has a higher amount of phosphorus in it, you're not going to be phosphorus limited, and this expression is going to take 1. If it has lower amount of phosphorus than you do, so Q is lower than theta, then you're going to be phosphorus limited, and your growth is going to be reduced by this expression here. So here are some simulations of their model under different light intensities. So light intensity here represents that carrying capacity K, carrying capacity in terms of carbon. So for low values of K, which represents low light intensity, here you have this dotted line is the grazing population over time. Excuse me, that's the producer population over time, the algae. The solid line is the grazing population, the Daphnia over time. And you can see under low light they have a steady state. Increasing light higher or increasing that carrying capacity K in terms of carbon, you see the classic predator-prey cyclings. The interesting case is when they increase K even farther under very high light intensity. You see the algae population, the producer, doing very well, going up here. But despite the high food abundance, the grazing population of Daphnia stays relatively low values and is unable to thrive. This is due to the fact that the food, even though there's a lot of it, it's low quality food because it has low amounts of phosphorus in it. So comparing this to that data is encouraging to see. They were able to include not just carbon, but carbon and phosphorus in the model now, and then capture dynamics that just thinking about only carbon in those models were unable to capture. Here's a phase plane analysis of their model. So here you have the producer or X population. Here's the grazer or Y population. The dotted curve here, that's the producer's null-cline. Should look familiar. That's the same kind of null-cline that we have in the Lotka-Volterra model with logistic growth. The grazer null-cline is given with this solid line. That has a new shape. If you remember from the Lotka-Volterra model, this is normally a straight vertical line. Now it has a bend in it right here. And that divides this phase space into two regions. One region where growth is determined by food quantity down here, like the original model, and this new region up here where growth is now determined by food quality due to low phosphorus in the food. So that was kind of the first part. We saw what happens when we have low amounts of phosphorus in the food. The LKE model essentially captures that by introducing a carrying capacity that's defined now in terms of carbon or phosphorus, depending on what's limiting, and the grazer's growth expression that's now going to be limited by carbon or phosphorus. The use of the minimum functions follows nicely from the ecologist Justin von Liebig's law of the minimum. If you remember what that is, the law of the minimum states that your growth is going to be limited by what other single resource in your environment is in lowest relative to your demands. Okay? But the next question is what happens when there's too much phosphorus? And this is a question I spend a lot of time working on over the years. Okay? What are the consequences when there are too much phosphorus in the system? Does it hurt your growth at all? And if it does, can we incorporate that into the model to make it even more accurate? Okay. So there's a lot of issues with our current phosphorus cycling today. What we're doing is we're mining a lot of phosphorus from phosphate rock here, and we're using it as fertilizer and animal feed. It ends up in our farms. A good portion of that ends up being runoff, like fertilization runoff, and that ends up polluting our aquatic ecosystems, such as lakes and oceans. So we end up having this phosphorus pollution over here in aquatic ecosystems, which might affect how the different, a lot of different aspects of the populations and how community structure of the systems. At the same time, many people are worried about that our levels of easily mineable phosphate rock are depleting really quickly. So we're actually limited on phosphorus on this end, and it's due to this kind of single directional flow. So there's a lot of issues with our phosphorus cycle, in that we might be limited here, and we won't be able to meet our fertilization demands to make the amount of food we're making now. At the same time, we have excess problems over here. There's a lot of research going on in this problem, a lot of great researchers working on different phosphorus recycling strategies. All I'm talking about today in my modeling is this aspect of the problem right here, where we have excess amounts of phosphorus polluting some of our aquatic ecosystems. Specifically, how that's going to be hurting grazers' growth and how we can incorporate that into our model to make better predictions about it. So here's some data on what happens to grazers when they get fed high levels of phosphorus. This is data from a lot of different animals here, and this is just to show you that this kind of phenomenon is observed in many species. I've been talking about Daphnia. This water flea down here was actually a Daphnia in their experiment. Okay? So on the x-axis of all these graphs, you have the amount of phosphorus in the food. So it's increasing phosphorus from left to right. On the y-axis of all these graphs, you have different versions of growth rate. Okay? They all have kind of the same shape. Over here, when you have low phosphorus in the food, you have this reduced growth. As you increase phosphorus in the food, the growth increases. That's something we've studied, it's kind of well known. That's phosphorus deficiencies, and the LKE model essentially captures that. It can limit its growth based off of low phosphorus content in the food. Things get interesting as you increase phosphorus, the growth continues to increase until at some point it reaches an optimal growth rate for some intermediate value of phosphorus. And then you see the growth rate starts to decline as phosphorus levels increase. And you see that in all of these organisms. The right-hand side of all these graphs dip back down. This is called the stoichiometric knife edge. And this phenomenon, again, is observed in all these species, where you have a reduced growth for low phosphorus content of your food, as well as for high phosphorus content of your food. Here's some more data collected by Plaza and Boersma in 2001 to help kind of explain why this phenomenon might be happening. So here, I have to apologize for these units. All afternoon I've been talking about phosphorus to carbon ratios. And this one right here is a carbon to phosphorus ratio, so it's flipped. But notice the numbers on the axis are also flipped. So you still have increasing phosphorus as you move from left to right. Okay? So here that they have algae of varying phosphorus contents, and then they fed that to Daphnia. Here, it's kind of hard to see the difference between squares and circles, but this curve right here has squares. This is the growth rate of the Daphnia. You can see it has that stoichiometric knife edge shape, where the growth of the Daphnia is reduced both when you have low phosphorus content in the food as well as high phosphorus content in the food. The other two sets of data you see here are the Daphnia feeding rates. They had two different types of food, but they both show the same dynamic, so we can just look at one of them. Under low phosphorus content of the food, notice that the Daphnia has really high feeding rate. And as the phosphorus content of the food increases, the feeding rate starts to decrease. And you have this kind of weird case over here where you have very high phosphorus content in the food, and they have a very low feeding rate. This promoted Plaza and Boersma to come up with a hypothesis to explain this phenomenon, that this reduced growth rate is a result of feeding behaviors. The Daphnia are reducing their feeding behaviors based on how much phosphorus is in the food. And on this end over here, they're simply not eating enough to meet their carbon demands, and that's depleting their growth rate. So high phosphorus content led them to decrease their ingestion rate, perhaps leading to insufficient carbon intake, and that would lead to the decreased growth rate. They called it eat until you get enough pee, then stop, as their hypothesis. I know it seems strange, but that's what this data kind of shows. You have this reduced feeding rate, and corresponds with high phosphorus as well as the reduced growth rate. So this is the hypothesis we used in our model. Taking the hypothesis that the Daphnia are going to be basing their feeding behaviors, they're going to be changing their feeding behaviors based on how much phosphorus is in their food. Okay, so here's the LKE model. Now we're going to adapt it to incorporate the stoichiometric knife edge. Okay? So the first thing we're going to change is the feeding rate. Right? We want to put a cap on that for when the phosphorus content is too high in the food. Okay, so taking the assumption that grazers are going to ingest pee required to the rate of its maximal growth, but not more. Okay? We're going to replace this feeding rate with what we're calling this uptake function. So this term highlighted in blue is going to be replaced by this new term defined down here, in order to put a cap on the feeding rate when we have high levels of phosphorus in the food. So there are two scenarios to consider. One where there's not excess phosphorus. That's the case when the ingested phosphorus is less than that cap we're putting on it. Here, F hat is the maximum of the feeding rate F. Okay? Then the feeding rate is going to take the original feeding rate from before and have the same feeding behaviors as the previous model. However, when phosphorus is in excess, we want to put a cap on that feeding rate. That's when the incoming phosphorus content is greater than that cap. We're going to redefine the feeding rate with this expression here. You can rewrite this as another minimum function. So now our uptake function is a minimum of two inputs. The original feeding rate for when when there's not excess phosphorus, and this new capped feeding rate for when there is excess phosphorus. Okay? So putting that term in there, our model takes this form. The next thing we need to rethink is the grazer's growth rate. So this term I have highlighted in green. Grazer's growth might be limited by phosphorus, but if phosphorus is in excess, grazer's growth is now going to be limited by carbon. So we want to incorporate that into the model. One thing I want to point out is Q. Remember, that's the phosphorus to carbon content of the algae, or the quota. You can see that's represented right here. But part of that ingested carbon ends up being used for metabolic costs such as respiration. So the phosphorus content of the post-ingested algae that's available for Daphnia growth is actually going to be higher than that. So you have this expression right here is actually the phosphorus to carbon content of the post-ingested algae that's available for Daphnia growth. So to think about this growth rate, we can take two approaches. one where there's not excess phosphorus and one where there is excess phosphorus. So when there's not excess phosphorus, that's going to be the case when that P to C ratio of the post-ingested algae that's available for Daphnia growth is less than the Daphnia's P to C ratio. Growth is going to be limited by phosphorus, and it's going to follow this expression right here, satisfy this equation. On the other hand, when there is excess phosphorus, that's the case when that post-ingested P to C ratio of the food is greater than the P to C ratio of the Daphnia. Grazer's growth is now going to be limited by carbon. And it's going to be satisfying this expression down here. So we have these two cases with these two different growth expressions. We can use the same kind of trick and rewrite this as a minimum function of the two. So you see that up here. Now our growth is the expression that takes the product of these two minimum functions. Remember the uptake function from before? The product of two minimum functions we can write as a single minimum function with 4 inputs, which we do right here. But one of these is redundant, and we can get rid of one of them because this inequality is true. E is the simulation efficiency that has to do with the metabolic processes. That's going to be less than 1. And F hat, again, is the maximum of F, so that's going to be E. E hat times F is always going to be less than the maximum of F. So we're down to this growth rate expression right here, the minimum of 3 functions. And that translates nicely into 3 biological cases. The first one is when growth is limited by food quality because phosphorus is low. The second one is when growth is limited by food quality because phosphorus is in excess. And the third one is when growth is limited by food quantity. So putting all the pieces together, we have the stoichiometric knife edge model. So we can do a similar phase plane analysis of this model. Here, X, again, is the algae population or the producer. Y is the Daphnia population or the grazer. The dotted curve right here, that's the producer null-cline. Has kind of a similar hump shape. The grazer null-cline is this solid line right here. And notice before, if you remember before, it was a vertical line here. Now we have this second bend in it. And that breaks this phase space into now 3 regions. The 2 from the LPE model, the green one right here where growth is limited by quantity, the blue one up here when growth is limited by quality because phosphorus was limited. And now we have this third region where growth is limited by phosphorus being in excess. Growth is limited by quality due to phosphorus excess. Okay. So we kept that assumption that the total amount of phosphorus in the system is constant. Remember that capital P from the model? So what we can do is vary P and see how the phase plane changes as we start introducing more and more phosphorus into the system. So you can see that happening here. This first graph is for low phosphorus. Then we increase phosphorus until we get to an excess phosphorus here. Okay? So you can see what's happening as we increase phosphorus. That producer null-cline, that dotted line, is being pulled down towards this bottom left-hand corner. It's being pulled down towards that red region. So under low amounts of phosphorus in this first panel right here, this is the only stable equilibrium inside right here. This is the solid circle. These open circles are unstable equilibriums. Okay? As you increase phosphorus, you're pulling, again, this dotted curve, the producer null-cline, down towards that red region. As you pull it down, these 2 equilibriums are going to collide, and you're going to have a saddle node bifurcation. And then you're going to see cyclones emerge right here. As you continue to increase phosphorus, you're continuing to push the producer null-cline down to that corner. A half bifurcation occurs as the interior equilibrium gains stability. And then here you start to see the effects of the knife edge, because as you increase phosphorus, you continue to push this null-cline down. This equilibrium is going to follow that line, and grazer density is going to start to decrease here as you increase phosphorus. To the extreme situation where you see in this excess phosphorus levels here, there's no longer any intersection, and you have grazer's extinction due to the fact that you have too much phosphorus in the system. Okay. So here are some simulations that correspond with those phase planes I showed you. Again, for low phosphorus, you have a steady equilibrium here. For intermediate values of phosphorus, you have those cyclones that occur. And then as you continue to increase phosphorus, you see it getting into that red region. And the extreme case over here, where you have very high levels of phosphorus, you have this this algae producer population doing quite well, but despite this high food abundance, the Daphnia population is unable to thrive and even goes extinct in this extreme case. This is due to the high phosphorus content of their food. We can look at these dynamics in a bifurcation diagram as well. So here's a bifurcation diagram using phosphorus as the bifurcation parameter on this axis, and here's the grazer's density, so the Daphnia population. Under low amounts of phosphorus, there's not enough to support a grazer population. As you continue to increase phosphorus, you do have a supported population here at this equilibrium. As you continue to increase phosphorus, you hit that settle node bifurcation I mentioned, and then you start cycling. Eventually, those cycles will collapse near this half bifurcation, and you'll have this steady state up here. And as you continue to increase phosphorus, the Daphnia population continues to increase until a point right around here. And this is where you start to see the effects of that stoichiometric knife edge. When this starts to decline, you have declining grazer density due to the fact that you have excess amounts of phosphorus in the system. In the extreme case, you can see under very high levels of phosphorus, these will even lead to extinction down here. Here's some theoretical curves. These are the knife edge curves made from the model. So these curves are made from our growth expression that we developed for the model here. So on this axis, you have the producer P content with a P to C ratio. On the x-axis, you have the grazer's growth rate. And there's not so much data out there that we know what these curves look like exactly, but we know that they have reduced growth both for low phosphorus as well as for high phosphorus. So we changed different parameters in our model to get an idea of how they affect the shape of these knife edge curves. Changing F hat, which is the maximum ingestion rate, changes the height of this plateau. Changing theta, which was the grazer's phosphorus to carbon ratio, changes the breadth of this plateau. And I am talking about extreme levels of, or not extreme, I'm talking about very high levels of phosphorus. So it's good to check to make sure this is actually a relevant problem to be talking about. So data collected by a lot of people, but compiled by Robert Sterner, of all the different algae phosphorus to carbon content ratios and a lot of different aquatic ecosystems. This looked at rivers as well as lakes in coastal regions. So they compiled a list of all the phosphorus to carbon ratios measured and found that up to 10% of them had levels of around 0.05. And you can see where that falls on this graph. It is in the region where you would have potential consequences due to this knife edge effect. Okay. So here are the assumptions of the model. I've mentioned them as I went through it. I'm not going to go through them all again. I just want to bring your attention to the third one right here. This is an assumption that was made by the LKE model as well, and that's that all the phosphorus is divided into two pools. It's either in the Daphnia or it's in the grazer. And this doesn't allow for there to be tracking free phosphorus in the environment. This assumption was based off of the fact that algae do tend to uptake the phosphorus it's given to them rather quickly. Okay? This doesn't hold necessarily in terrestrial systems, where nutrients might spend more time in the soil than they would in an aquatic system. Okay? Also, this might present a problem because I'm talking about very high levels of phosphorus. It might not be biologically possible to fit all that phosphorus in your algae population. Imagine if you had a very small algae population and a high level of phosphorus. The model right now assumes all that phosphorus is in the algae population, which means it has a very high quota, like P to C ratio Q, has no upper bound on it right now. And it might not be biologically possible to have the algae with that high of a quota. And if we let that happen, it almost becomes like a suicide pill for any Daphnia that comes and eats it, because it eats it and then automatically it reaches its phosphorus demands, reduces its feeding rate, and then will starve. So in order to put a cap on this Q, put a cap on our phosphorus to carbon content of the algae, we have to track the nutrients in the environment. So we extended this model to track the nutrients in the environment. And you can see that model right here. Okay? So these first two questions are the same. The first one looks a little differently, but it's the same as it was before. Here you have the population of algae. Here you have the population of Daphnia. And I have two new differential equations. One's the quota, or the phosphorus to carbon ratio of the producer, Q. The second new equation is the amount of free phosphorus in the environment, PF here. This function right here is the phosphorus to carbon uptake rate of the algae. I'm going to come back to talk about that in a little bit. Using the assumption that the total amount of phosphorus is fixed, that capital P, we're able to reduce the systems of 4 ODs down to 3 ODs. So we can get rid of that last one, look complicated. So now we have a system of 3 differential equations, one of the algae, one of the Daphnia, and one of the algae phosphorus to carbon ratio. Okay. Now I'm going to mention more about this uptake function here, V. Okay. The uptake function takes 2 inputs. The first one is the amount of free phosphorus in the environment. The second one is the amount of quota, the phosphorus to carbon ratio of the algae. And that takes this form right here. So there's 3 new parameters introduced into this model with this function. The first one is Q hat, the maximum phosphorus to carbon quota. That's the cap we wanted to put on the quota, so that's good to have here. The other 2 is this C hat and this A hat. The C hat is the maximum phosphorus to carbon uptake rate, and A hat is the half saturation constant for this portion right here. So I'm going to come back to C in a little bit. So just remember, C is the maximum phosphorus to carbon uptake rate of the algae. Looking at some simulations of this model for the same different level of phosphorus. Under low levels of phosphorus, you do have equilibrium values. Now we have 3 equations, so we have 3 solutions. This top dotted or dashed line is the algae or the producer population. This solid one is the Daphnia or the grazer population. And this new line, this new set of data, this dotted line, is the producer phosphorus to carbon ratio. You can see under low phosphorus levels, it's at equilibrium here. As you increase phosphorus, you do have cycling that will occur. And then this extreme case over here, where under very high levels of phosphorus, you see the Daphnia population, this dashed line right here, at high levels. Notice it also has a very high phosphorus content. You see the Q or the P to C ratio up here. And due to that excess amount of phosphorus in the food, the Daphnia population is down here and is unable to thrive. So we can take a similar approach where we look at the phase space of the model. It's a little bit more complicated now because our model is 3D, so our phase space is going to be 3D. Instead of looking at null clients, we're going to be looking at null surfaces. But we're interested in changing that parameter P, the total amount of phosphorus in the system. And we got kind of lucky here, because if you look at these 3 equations and where you see a P, it's only in this last expression right here. Okay? So when we look at these null clients, these null surfaces, and we change the value of P, the Q null surface is the only one that's going to change. So we have these 2 surfaces fixed, and we just have to look at how one surface sweeps through that area, that phase space, and changes. Okay? It's similar to the 2-dimensional model I showed you. Remember when we increased phosphorus, all that ended up doing was pulling the dotted producer null client down into that red corner. Here we're just going to be doing a similar thing where we're shifting the Q null surface. Okay, so you see that here. The yellow surface here is the grazer surface, Y, null surface. You see under different phosphorus contents, it's in the same, it doesn't change at all. Okay? The blue surface is the producer null surface. It's also staying constant across these 3 graphs. And what's changing is the red null surface. The red null surface is the Q surface, or the phosphorus to carbon ratio of the algae. And you can see how it changes. For low amounts of phosphorus, it's down here. As you increase phosphorus, this red plane sweeps through this space until it ends up here. Remember that equilibriums occur when you have the intersection of all 3 of these planes. So we were able to look at the intersection of all 3 of these planes, run simulations, and see when they were stable and unstable. All that work's kind of summarized up also here in this bifurcation diagram. So here we have the grazer's density on this axis Y, and we have total amount of phosphorus on this lower x-axis here. It has a similar bifurcation structure to the one you saw in the 2D model. At first, when you have low levels of phosphorus, it's unable to support grazer's density. As you continue to increase phosphorus, you are able to support grazer's density. It'll continue to increase. There'll be a saddle node bifurcation, and then cycling will emerge. You'll have cycling for a while. They eventually collapse near a half bifurcation. Then you'll have a stable equilibrium again, which will eventually start to decline due to the effects of the stoichiometric knife edge, and eventually lead to extinction for very high levels of phosphorus. We also found 2 regions of bistability here, one around the half bifurcation. It's a very narrow window. You can see it blown up here. And one around the second saddle node bifurcation, another kind of very narrow window, but you can see it blown up right here. Then we can compare the bifurcation diagrams of our 2 models. The 2D model here is on the left, the knife edge model. The 3D one, which we call the full knife edge model. The full model is here on the right. Okay. So it has similar bifurcation structures. You hit a saddle node bifurcation, cycling hop bifurcation, and eventually another saddle node bifurcation. But there are some differences you can see here. The biggest one is the amplitude of those cycles. So looking over here, you notice these cycles have quite large amplitudes, and the Daphnia population is spending a lot of time down here kind of towards dangerously low values where they're endangered from stochastic extinction. Okay? Also, the placement of the hop bifurcation is different. Here it's occurring at around 0.06 milligrams per phosphorus per liter. Over here it's occurring at, pushed to the right at around 0.08. So the length of these, the period of cycling is also longer. Okay? The period right after the hop bifurcation, when you have this stable equilibrium up here, this is what I like to call the safe zone. If I were a Daphne, I'd want to be living here. Here you have this high grazer density, but you're not in a cycling regime here. So notice this kind of safe zone right here is a lot longer than the safe zone over here in the full knife edge model, where we have a cap on that quota and we're tracking nutrients in the environment. So here's some data collected by Dodds and others in 2008, where they looked at the different phosphorus levels in a lot of lakes throughout the United States. There were some that were actually quite high, especially in the Great Plains area. So in the Great Plains area, you had phosphorus contents of around 0.6 to 0.8. So if you look at the two different models and try to decide which one to use, here if you were using this model and you had 0.8 for your phosphorus levels, you'd predict a pretty high grazer density. Whereas over here you had 0.8, you're still in the cycling regime where you have these high amplitude cycles and you're in danger of stochastic extinction. So it's quite different predictions. Now I did tell you that we introduced 3 new parameters when we made this model here. And those 2 parameter values do have an effect on the bifurcation diagram. Okay. So the Q hat and the C hat were the important ones. Turns out that the 2-dimensional knife edge model is limited in case of the full knife edge model when these parameters go to infinity. That makes sense because remember the assumption of the 2D model was that it didn't allow phosphorus to be in the environment because algae uptake it really quickly. Uptaking it quickly is the same thing as C going to infinity. Also, there's no upper bound for the amount of phosphorus in the algae, so letting Q go to infinity as well. Okay. So here are 2 simulations of the bifurcation diagram of the full model for different levels of C, the carbon to phosphorus uptake rate of the algae. When it's 0.2, you have a bifurcation diagram like similar to the one I showed you earlier on the previous slide. When you increase that to 0.8, notice the half bifurcation was pushed to the left. So it's looking closer like the 2-dimensional knife edge model. Okay. As you continue to increase C, this will converge, NQ, this will converge to be in that model. So which model you use is going to depend on your parameter values. If you can measure C hat, the phosphorus to carbon uptake rate of your producer, and it is high, you might be able to get away with using that 2D model rather than this complicated 3-dimensional model. Okay. So ecological stoichiometry has a lot to offer. I certainly think this is so, and hopefully I've convinced some of you that this is true. We saw how we took a single currency model that only considered phosphorus. We introduced phosphorus limitation into that model with the LKE model, and then some work with the knife edge where we introduced effects of excess phosphorus. Okay, we saw that their dynamics made quantitatively and qualitatively different predictions for the ecological ecosystems we were looking at. So you can see how this theory, ecological stoichiometry, has impacted models over the years pretty clearly, I think, with looking at the phase spanes. So in 1920, we had the log of Volterra model here. This is before they had logistic growth in it. And you can see the dotted line here is the producer null-cline. That was just a straight horizontal line. The grazer null-cline was a straight vertical line. In the 60s, they introduced logistic growth. So they have this newly formed producer null-cline now with this hump shape. And then in 2000, you start to see the influences from ecological stoichiometry in considering food qualities. Here in this model, they considered low nutrient quality in their food. And you can see the grazer's null-cline now has a very different shape. It has a bent here that divided this phase space into 2 regions. One where growth is limited by food quantity, one where growth is limited by food quality because it's low phosphorus or low nutrient content of the food. Now introducing this knife edge models here, we've changed these null-clines again. You can see the grazer null-cline has an additional bend, dividing the phase space now into 3 regions. This middle region where growth is limited by food quantity, and now 2 regions where growth is limited by food quality. This one up here, growth is limited because phosphorus is limiting. This one down here, growth is limited because phosphorus is in excess. Stoichiometry can also lead to some non-intuitive paradoxes that have been used, you can see them in these models. So first, just to recall, Rosenberg and MacArthur had that paradox of enrichment, which you can see right here at this Hoff bifurcation, if you have this system and you enrich it, you'd move to the right a bit and you'd start to get into a cycling regime. So it destabilizes this equilibrium here. That was called the paradox of enrichment. Looking at the LKE, there's a new paradox we see here. The paradox of enrichment is still there at the Hoff bifurcation, but farther on, as you continue to increase, in this case, they increase light intensity, you start to at the saddle node bifurcation, you start to see this decline in Grazer's growth as you increase the carrying capacity K. So they call that the paradox of energy enrichment. It's kind of non-intuitive behavior, whereas you continue to increase the amount of energy going into the system, you start decreasing the Grazer's density. Now we have another paradox in the knife edge model. Here you have phosphorus now as the bifurcation parameter instead of the carrying capacity. So we have the same bifurcations, they just occur in the opposite order. So notice it goes this direction here. That's because phosphorus, that's kind of the food, increases from left to right in our graph and decreases from left to right in their graphs. Okay? So the same bifurcation but in an opposite order. You have this region right here where it exhibits the paradox of energy enrichment. You have the paradox of enrichment here. And now this new paradox, when you see the effects of the knife edge, when you further enrich the system with phosphorus, you actually start to hurt the Grazer's growth, and you see their growth beginning to decline. So we call that the paradox of nutrient enrichment. So some work I'm going to be doing here. The big one is developing a food web model. So kind of expanding upon these producer-grazer model to incorporate a third trophic level. So I can look at answering questions about how nutrients are flowing through the trophic levels and up the food chain and maybe accumulating in the third trophic level. Second, I think that ecological stoichiometry has a lot to offer to ecotoxicology. And asking questions like whether or not, considering stoichiometry can help improve testing protocols for assessing risk of exposures. So for the food web model, this is the very first iteration. I'm sure this will change as I continue looking at this. But here, this is the first one. We have the producer population, a grazer population, and now I introduce a predator population, a predator that's predating on the grazer here. Okay? So after continuing to work on this, I do think we can look at how nutrients are trophically transferred between these three different trophic levels, and if they accumulate in any of them. How will the addition of this predator change the population dynamics and community structure of the system? And then I can look at what roles see maybe consumer and predator nutrient recycling rates play and total nutrient level availability. And how will that change population dynamics and community structure? After we have a good food web model, I think we're sitting in a pretty good spot to start looking at some applications in ecotoxicology. So we know that nutrient stressors, I've been talking about that food quality issues a lot this afternoon, they have an effect on life history traits such as growth and reproduction. We know that toxic stressors have the same kind of influences on this life history trait. So I think this framework would be a nice place to start looking at co-occurring nutrient and toxic stressors. Life history traits that get varied like this affect population dynamics and community structure, as well as the trophic transfer of nutrients and potentially the trophic transfer of toxics as well. Okay? So instead of just looking at how the question of how nutrients are transferred across the trophic levels, we can look at how toxins also transfer up to trophic levels and whether or not they bioaccumulate somewhere. And I think models like this will help us determine the importance if food quality should be an issue to be discussed for assessing risk for exposures and water quality policies. So just to conclude, theory of ecological stoichiometry is a powerful tool. Again, I hope I've convinced some of you of that. Including food quality and multiple currencies can help improve models. We see the difference both qualitatively as well as quantitatively different predictions we see with both the LKE model and now the knife edge models. Developing a food web model like this, I think, will only further our understanding all these food quality issues. And again, I think this framework has a high potential in ecotoxicology applications. Okay. Lastly, I want to say thank you for everyone for being here. And I was so jealous of Liz's beautiful parakeet photos from her talk. I thought I could include some pictures I took myself at the zoo. So Thank you.